Okay, thank you very much for this invitation. I must say that during the last almost 10 years, my research has been funded to a large extent by CRCNS. I got 3 grants, CRCNS, with various collaborators. And today I'm going to talk about one of these works that we did in collaboration with Nicholas Priebe, an experimentalist, Carl von Weisweig, who passed away in 2020 unexpectedly, my very close friend, and Herman Matteau from Bariloche, Jaguti Patatkal from Austin. So first of all, so it's dedicated to the memory of Carl. So it's well known that in the visual pathway, so orientation selectivity emerging V1, and for many, many years, the question has been how this comes in. So just to remind you, so this orientation selectivity responds to a visual stimulus, which is a drifting grating with orientation theta, spatial frequency lambda. And so as you see, the neurons respond preferentially to stimulus orientation, which, the response-dependent stimulus orientation, we call preferred orientation, the orientation of the stimulus at which the response is maximum. So it's well known also that in cat V1, as well as in monkey, ferret, and quite many other animals, so there is an orientation map in cortex. That is, it's ordered, yes, which means that neurons which are close by have similar functional property, and in our case, we're talking about preferred orientation. So the preferred orientation is encoded with a color code. So this is so as you see, so neurons are very well ordered in terms of functions. So now there are some, for many years also, the question has been, what is the mechanism? And I call this the power of order. And it's the mechanism basically that we believe now to be the case in cat V1, and in other case, in other animals where there is an orientation map. There are 2 contributions. One contribution which comes from the thalamic projections to V1. They are organized. This is a Hubel and Wiesel idea or mechanism. Convergence of multiple untuned LGN input generates some selectivity in the excitation of cortical neurons. This excitation of cortical neurons coming from LGN, I will call it later aggregate thalamic excitation. The other component is due to the recurrent connectivity in cortex, and this recurrent connectivity is, as I said, everything is ordered, is feature specific, and it's believed that this, the cortical dynamics sharpen the selectivity. So the input coming from the thalamus is already to some extent tuned to orientation because of the organization of the projection and because of the feature-specific connection, the cortex is sharpening the selectivity. What happens in rodents? So in rodents, in fact, it's not also for quite many years that there is disorder. There is no map. It means that neurons which are close by can have very different preferred orientations. This is again shown here in a color plot. So the question is, could random connectivity, so we are going to take the extremes that the cortex of rodents is, the thalamus cortex network in rodent is disordered. So we assume random connectivity, and the question is, can we account for mass V1 orientation selectivity in this setting? So so that means that here, what we assume that the thalamic projection to V1 are not organized. There is random convergence of multiple untuned LGN input onto layer 4 neurons. And moreover, the recurrent connectivity in cortex is random, which means that cortical neurons integrate recurrent inputs from neurons with, other cortical neurons with random preferred orientation. So here what we did to address this question, could random connectivity account for mass V1 orientation selectivity, our approach goes as follows. First of all, we have an analytical solvable mean field theory done earlier, attacked this kind of, was quite critical about this kind of approach, and I disagree with him in this aspect. So we assume in that case a large number of thalamocortical and intracortical recurrent inputs. So this gives us a first idea of the mechanism, how it can be that in a network with random connections, there is orientations, there is selectivity. Then what we do simulation of sparking network model of the LGN-V1 network. So this is in order to test the robustness of the result that we have achieved in our theory, in our mean field theory, robustness with respect to the properties of the parameters of the network, and which, of course, now it's not, connectivity cannot be very, very large in reality, and the robustness to other parameters. Then we do validation. We compare with established experimental results, then predictions, and again, validation of these predictions, and more predictions that have to be validated. So we first of all consider the aggregate dynamic excitation, which means the input coming from the LGN, so total input, the total excitation coming from the LGN to a neuron in cortex. And here again, we are dealing with drifting grating characterized by some spatial frequency, temporal frequency, and we have two critical parameters here. One is the dispersion of the connectivity, sigma, so which characterizes the spatial structure of the probability of the connection to a neuron in cortex. So again, it's random. We assume that there is randomness. And K, which is the number, the average number of inputs that a given neuron in cortex receives from the LGN. Now, in experiments, there is some experiment which has been performed in Scandiani 2013. They were able to measure the selectivity properties of this aggregate dynamic excitation. So what our theory predict? Our theory predict first that the ATA0, this is the F0 component. So this is the average in time of the aggregate dynamic excitation. It isn't tuned. The experiment shows that, so Lin and Scandiani found this, can characterize, quantify this degree of selectivity in terms of what is called the orientation selectivity index, and they found 0.03, which means that's very weak tuning. Now, our theory shows that the F1 component, which means that the modulation, temporal modulation as a frequency, temporal frequency of the grating is tuned. More precisely, what we show that the degree of tuning depends on the ratio between sigma, the dispersion of the spatial dispersion of the connection from LGN to the cortex, over lambda, the spatial frequency of the stimulus. Now, what the experiment gives, so the OSI of the F1 component is 0.23, and this is for 0.04 cycle per degree. So now we can go into our curve and show that here we have 0.23, and from this we can use that and the prediction that the dispersion, spatial dispersion of the connection from LGN to cortex is 7 degrees. Now, there is another property of our theory that we find that the on and off subregions of the thalamocortical inputs overlap. And in fact, in experiment, this is found. So again, this is in the paper of Lin and Scandiani, 2013. And what they find that the offset of this on and off region on average 5 degrees. Let's go back now to the theory. So what we find that as a function of the number of input that a cortical neuron receives from LGN, so the mean offset varies. And if we go back to the experiment, so we want to have 5 degrees, as in the experiment, and we find that on average, LGN cell receives 20 input from the cortex. And there is another way to estimate this number of input. It is by looking at the ratio between the F1 and the F0 of the dynamic excitation. And so what we find that again, by comparing to the experiment, we find that in K is equal to 20 inputs on average per from LGN to cortical neuron. So this is in a paper, which is what we call in submission, thalamocortical connectivity in rodent V1 is random. And this, we have been working on this over the phone during all this, the confinement with Carl von Vreeswijk and our collaborator, and we are still working on it. So now what happened in cortex? So in cortex, we find now that this input, which conveys some information about the orientation of the stimulus, give rise to selectivity also. And here also we can compare with experiment. Now what we find that in the cortex, we have indeed a salt and pepper organization. Now we can go further, we can compare quantitatively with the degree of selectivity in cortex in experiments, and this is what is shown here. In blue, this is the experiment, and in gray, this is our model. So the model shows that what is necessary for this is that in cortex, the recurrent excitation, the recurrent interaction, both excitation and inhibition must be strong. So in other words, this is a famous balanced state, and this shows that there is a lot of advantage to have balance of power. And this is also true in politics. Now the question is this verified by experiments? So there is a lot of work on that. It's not whether the cortex operates in balanced state is still a matter of debate. Ram Darshan is a specialist of this question, testing this hypothesis on data. The prediction of this, of our theory is that the preferred orientation depends on spatial frequency, and here we verify this prediction. Actually, it's in the lab of Nicolas Privé with his postdoc Jagoti Padarkar, verifies this prediction. This is something which is very different from what happens in cat and monkeys. There, the preferred orientation is very stable when changes spatial frequency. This is published in a paper which was funded by CRCNS. So this is a paper, you can find much more details there. So the conclusion. So what our work has shown that for the thalamic excitation, the observed orientation tuning and receptive field properties can be accounted for with random LGN projections. So thalamic excitation gives rise to orientation selectivity in cortex. Predictions are verified. Preferred orientation of V1 responses changes with spatial frequency. What I did not show that there is intricate to the orientation spatial frequency receptive fields. There is a prediction to be verified K equal 20, and in fact, we have much more predictions that I did not have time to present here. What I did not show is how layer 2-3 now inherits orientation selectivity from layer 4. Our theory also can explain direction selectivity, how it emerges from diversity in LGN dynamics. And finally, we can show that a few strong specific connections as observed in experiment, as seen in physiological and anatomical experiment, can sharpen orientation selectivity. All these things are paper in preparation. I would like to finish by advertising the Van Treewijk Theoretical Neuroscience Seminar, which is on Zoom, every Wednesday at 11 a.m. And we are, next year will be the 4th season. We founded it with Carl von Vreeswijk, and the seminar now as his name. And finally, acknowledgements. So here in red, those who are contributing to this study, and in bold. So these are those who are we have been associated, collaborating in the different CRCNS grants that I have and we have benefited. Thank you very much, and I see there was 3 like this. Thank you.